Live from the Orion Cygnus arm of the Milky Way galaxy, this is Scientifically Speaking, a weekly half-hour program devoted to elegant curiosities. And I am one of your hosts, Sarah Chang, and joining me, as always, is Bernie Rhyme, DJ Star Watcher. How are you, Bernie?
Yes, hi Sarah, very good, thanks.
And Bernie is our professor of the astronomy lab at USM and the author of the monthly What's Up column in the Portland Press Herald. And also Skylight, the official newsletter of the Astronomical Society of Northern New England. Welcome back to another wonderful Friday.
Yes, it's Friday.
And reach out to us at WMPG, scientifically speaking, at gmail.com or on Twitter at WMPG You can ask questions or tell us how much you love our show, how much you love listening to our voices. That would be great to hear. If you've got questions, go ahead and throw them at me. Bernie, what's up in the night sky for this coming week?
Yes, I'm glad you asked. We have a good week coming up, probably not quite as exciting as last week, but you'll still be able to see a few leftover Perseid meteors. The moon will be waning crescent, so it'll be out of the way. It's not going to come up till about 3 a.m. So I saw some Perseids the other night. It was a pretty nice night. It was a little humid. I saw 8 meteors in just over an hour. I had a nice field of view. You know, I was out by a golf course, so I was kind of lucky. 3 of them were really bright, and then there were a couple others that were pretty good. It was strange, too. The first meteor went right near the Big Dipper where the comet was. I was looking for the comet, but it's getting a little too faint to see without a telescope, so I couldn't really see the comet. But it's still out there. And then 2 planets were already up. Jupiter is the bright planet. It comes up before sunset. I got down there about 10 o'clock at night. And Saturn is about 10 degrees to the left of Jupiter. It's not quite as bright. It's kind of a golden color. And then you had to wait till about 10:30 or so for Mars to come up. Mars is getting really bright and orange and very close to us. And if I would have stayed till 3, which I didn't, then Venus comes up around 3 in the morning. So the moon, when I saw it a few days ago, came up right at midnight, so now it would be 3 in the morning. But it was really nice. It was an orange last quarter moon, and you could see it just coming up above the horizon, deep orange color, and that pretty much washed out some of the rest of the meteors. So you can still catch a few meteors, and you can still look for the comet, but you would need a telescope, and you have to know exactly where it is. And then you can see 4 planets. Oh, when I checked my little phone app, between Jupiter and Saturn, Pluto is actually right in between those 2 planets now. Of course, I couldn't see it without a telescope. I did see it once, but it's kind of neat to know that Pluto is right between Jupiter and Saturn when you look in that part of the sky in Capricorn areas. You can see that. So that's coming up for this week.
That was my dog.
It's your dog.
Actually, Bernie, I have a question about the planets. Can we? I don't think I've ever seen Neptune or Uranus. Are those visible by naked eye?
Oh yeah, I've seen them many times. We saw them, I think, on some of the Acadia things too. I think some of the scopes had them.
Really?
Neptune is kind of bluish. It's like 8.6 magnitude, so you need a telescope. And Uranus is kind of greenish, and you can theoretically see it with binoculars, but I've never seen it without a telescope.
Wait, so you can't see either of them with?
You can't see either one naked eye, no.
Okay. Okay.
So I usually don't mention them when I talk about just the 5 bright planets.
Right. Right. Okay. Cool.
Yeah.
Thank you, Bernie.
Are you welcome?
So if Bernie talked way too fast, you can also check out the monthly What's Up column in the Portland Press Herald and read all about what's up in the night sky for the month. Bernie, could you please do us the honor of introducing our esteemed guests?
Yes. Yes, we have some interesting guests on this week. I've known both of them before. They've taken my astronomy class with me when I was teaching that course at the New School in Kennebunk, and they're both college students now. So we have Xander Davenport, who's studying biology at Hampshire College, and then we have Payce Shepherd, who's studying English at Wheaton College. So I've known them, and I asked them to be on the show. We were going to do a different topic at first because they have a wide range of interests. But so we're going to talk about a certain thing that's very much related to what's going on with the coronavirus now. These are called retrotransposons. So we're going to talk about them and see how they relate and get people more interested in the possibilities and those type of things going on.
Thank you, Bernie, for that warm introduction. So yeah, as he said, we're going to be talking about transposons and how they work. It's a sort of a not well-known topic that's really interesting, and we thought it would be nice for everyone to hear about. So basically, humans are full of DNA. It's like a script, okay?
Oh, dear.
And it's used to make who we are and how we're doing. But the thing about DNA is most of it isn't used specifically for the things that DNA is usually used for. It's sort of like what people used to think it was sort of like broken script or script that just isn't used at all. But now we realize there actually is some point to it, and it is affecting us all the time. And then transposons is one way it affects us. Now, what transposons do is it takes one of these DNA strings, think of it as a string, right? And it takes a part of the DNA and then moves it to another part or another string of DNA, right? Yes. And that's called mutation. Now, we don't generally like the mutations because often they aren't great because they're random, but every once in a while, a mutation will happen that's good. That's how things evolve, people are thinking.
Why does it? Why is there a necessity for it to move parts around? Because I think we understand DNA as, there are certain sequences in DNA that are expressed in certain, not characteristics, but I guess traits, right? Why is it necessary for it to want to move parts of them, and how does it decide what parts are moved around?
So what's really interesting, so a transposable element isn't actually moving parts of it itself is a sequence of DNA that is capable of making a copy of itself and then inserting that copy back into the genome at a different location. And it is really interesting because it could almost like be considered like a parasite in a weird way, where like even though it's contributed a lot to evolution, it is arguably acting more like a virus and that it's interested in its own replication more than it is in like a positive outcome for your genetic structure as a whole or like the organism in which it is present.
Functions like a retrovirus.
Yeah, a lot of them function similarly to retroviruses. It's a particular class, and they're kind of tied to retrovirus evolution. So the way that it basically works is you have a unit of DNA, which is basically capable of making a copy and then pasting that copy into a different section of genetic code.
It's mutation.
Yeah, which is a form of mutation. So there's a few different kinds and classes of them, but basically all of them fit into that same general idea where you have one, and then when it's transcribed, that copy is then reverse transcribed back into the DNA strand somewhere else.
So is a transposon a sequence of DNA, or is it a protein or something that does the copying?
It is a sequence of DNA, and many of them, though, code for the proteins that reinsert them back into the primary transcript. That kind is classed as an autonomous retrotransposon, which basically means that within its sequence, it encodes for the proteins that will bind to the RNA that's transcribed from it and facilitate connecting that RNA to a chink in DNA that it makes, and then translating DNA from that, which is basically the thing that, yeah. I kind of first came upon them and thought that they were interesting because of this thing that I heard about called horizontal gene transfer in prokaryotic life forms. So both like any kind of bacteria or archaea, they are able to take sections of their, so they basically have all of their genetic information in plasmids, which are just circles of DNA. And some of these plasmids, they can share with each other. So if one bacteria has a gene that codes for something like resistance to cold, or like resistance to an antibiotic, or resistance to heat stress, it can make a bunch of copies of that particular DNA sequence that has the gene for whatever that trait is that's really advantageous, and it can give it to other bacteria that are around it so they get that same trait. And it's really a different evolutionary process than what's happening in most eukaryotic life forms like humans or plants or anything else that's like a large multicellular organism, because our genetic process historically has been, in terms of how we evolve, has been thought of as this very kind of generation to generation thing. Like an organism gets a mutation, it passes it on to its children, they pass it on, and it's kind of held within the scope of a line. And it's where with of a line of one generation to the next, where with bacteria they can take their DNA and if it's a mutation that if they have a gene that they like, they can share it basically. And so what's
with
really
other bacteria.
With other bacteria and archaea. Yeah, sometimes. Actually, I don't know about archaea. Sorry, I shouldn't throw that in there.
So if I can dumb this down even more, if let's say, Xander, you really liked Payce's hair, and Payce was like, "Yeah, I'll just share that DNA sequence with you."
Yeah, and then I would have some of Payce's hair and my hair together, and it would be a wild party. Yeah. So and so it's a really cool process, and it's like an almost a totally different way of looking and thinking about evolution, because it's like, evolution's always kind of thought about as this thing of competition, and you've got to beat out the other guys, but like in a way it's like sharing is caring, kind of, like everybody's giving each other their genes, and like, oh, you have a gene I like, thank you, I'll have
but
one.
then, so is it possible that a bacteria can be like, hey, do you want this, this particular DNA sequence that'll give you this particular trait, and it's actually really bad?
Yeah.
That's a really good question. I actually don't know the answer to that. It's probably true.
I assume that happens. I mean, how would a bacteria know that something is beneficial?
I mean, it wouldn't necessarily. The thing about bacteria is that they're multiplying really fast. And so if they get something, they're able to kick out genes at will. So if there's something that's not being beneficial, they're kicking them out and taking them in all the time. And if one kicks one out that's not good for them, and it's like bad, then they'll either, their replication will slow down. And if another one keeps it, then the replication of that one will be outcompeting it, kind of, So it's like, it is a selection game in a way, but like they can kind of, constantly, just pass it around, kind of. And, they get rid of them really fast. If like, for example, it's a really good question though, because I imagine that you could have a plasmid that codes for a poison that one is immune to. I don't know if that happens or not, to be honest.
Yeah.
But
I guess evolutionarily speaking, it's not helpful, because then everybody would die.
Yeah.
Yeah. I think what we were talking about before was you need a species with enough of it to evolve for the times, but not too much of it to mutate out of existence, right?
Yeah, that's just kind of with just like general mutation.
Yeah. Okay.
But I think so anyways, this kind of ties into retrotransposons in a weird way, because and this might be like too big of a subject thing, but kind of, so what got me interested in them is that this kind of concept of one organism giving another organism a gene was something that was really thought to only happen in bacteria and archaea for a really long time. And also some single-celled eukaryotes, so like amoebas and stuff can do it. But like, nobody would ever think like, oh, there's a tree and there's a corn plant, and the corn plant might give the tree just naturally a little bit of its DNA. Like that is not something that, was really toyed with or thought about as a possibility, because it's kind of a wild idea in a way. And so what first got me interested in transposons was this idea of horizontal gene transfer in kind of higher eukaryotic organisms, like multicellular organisms that reproduce through sex. And basically, it looks like, from looking at the genomes of like lots of different species, it looks like sometimes one of these little bits of DNA that's able to kind of make copies of itself and cut and paste itself back into one organism's genome can sometimes hop to a different organism, and it can be really random, the crossovers. And what's interesting is people think that it's happened basically by looking at like sequences and being like, there's this repeating sequence that's happening a lot in this organism. All of a sudden it starts happening over here. But like kind of how that process is happening and how that they might be jumping from one organism to another isn't super well elucidated. There's a lot of theories as to like how that it might be happening. And there's also a lot of debate as to the extent to which it is happening, because like some people argue like the number that we've identified, which is not that many, of cases where that, a quote unquote jumping gene or like a transposon might have moved from one organism to another, might be kind of the tip of the iceberg in the amount that it's happened. But it's also kind of, that's just like purely hypothetical as well. Yeah.
I'm curious what that has, that kind of mechanism, does that have implications for genetic engineering in a way? Like, how could you take advantage of that? Is there a way to take advantage of that? Have people thought about that at all?
I think Payce has. Yeah.
Wait, so Payce, you have thought about that?
Yeah, we've thought about that.
Yeah, we have.
Mm-hmm.
Yeah.
But I'm not really sure if anyone else has.
I think I wish that I knew more about the subject to be able to like give you an answer on that, because I think that there's some forms of genetic modification that use processes that are kind of similar to viruses or viral infections, which can be really closely related to this.
There are some examples in real life, like what Xander told me before. Like there was in London during the Industrial Revolution, there was all the trees became black because of the soot.
Yeah,
right?
The birch trees.
Yeah, the birch trees. And then there were these moths that evolved to turn black to camouflage with the trees. And then
they used to be like speckled white, like the birch bark.
Yeah.
Mm-hmm. And that's as a result of that's as a result of retrotransposons?
Yeah, that's what people are thinking, yeah.
Well, they found when they studied the mutation that it was a retrotransposon that caused that mutation. And kind of why I thought it was really interesting is like this example, I wish that I had in front of me the name of the kind of moth, but it's an example that was like a pretty early thing that was looked at, a piece of evidence for the existence of evolution and the study of evolution as a concept, because this is like, the late 1800s. And so it's just kind of, and it was used in textbooks forever. Like even in my, high school biology class, we talked about like this moth that, we talked about like the moth that like changed with, with the change in tree color.
Is it the peppered moth?
It is the peppered moth, yeah. And so, anyway, and so like just, I think that people like who are like really nerdy about transposons found that out and were like, look, it's everywhere. The best, like most classic biological example out there is a transposon.
Is this something that could happen with a species as complex as us? I mean, not to say that butterflies are not complex, but
I think kind of what I realized, I might have confused two different things a little bit. not confuse two different things. So it can happen in something as complex as us, and humans actually have two active transposable elements in their genomes. And the rate of transposition, so like the rate of somebody having a random mutation due to one of them replicating, is relatively, it's not like, so let's see, it's like, it's like, it's under 1 in 200 live births, there's one. And there's two, so for the two different retrotransposons, they're pretty different. They're pretty different in like their rates in humans. Yeah, they're called long interspersed element 1, and or which like line 1 for short, and then ALU. We don't really know whether or not they come from what I was talking about with horizontal gene transfer. They're just kind of endemic to our genomes and are kind of continually replicating within us, very slowly, and probably eventually will just evolve into eradication, but currently they're both still active.
So evolve into eradication, are you suggesting that we had more?
We definitely had more. Yeah, and we actually have evidence of a bunch of them that are just no longer capable of functioning.
Is that just evolutionary? Like I guess, like with the butterfly example, like with the horizontal gene transfer, that seems like a very beneficial thing in some of the examples that you've shared. Are retrotransposons, are they, and I know you kind of described them as viruses, are they always bad?
That's a really complicated question. I do want to clear up one thing, which is that the mutation in the butterfly was caused by a transposon, but I think it was a horizontal gene transfer event, because like those two things can be a little bit separate,
Okay. So with the butterflies, it's likely it was generational.
Well, it's likely that it was, so it was generational, but it was caused by a transposon within a butterfly, So it wasn't that transposon, it was a transposon replicating and then reinserting into the butterfly instead of somehow leaving that butterfly and ending up in a different species,
Yeah, makes sense. I guess when you talked about that change,
I know
it sounded like it was, in a month, but it was probably a longer time frame.
Yeah. I don't know enough about the history of that particular organism to know how long it took. In terms of like if they're good or bad, I think it's kind of a complicated question, because it's like kind of the question of, is mutation good or bad, because like all organisms need mutation in order to keep evolving, but also at the same time the probability of having a beneficial mutation is really small in comparison to the probability of having a negative mutation. Pretty much with transposons in general, the danger of them to an organism is causing too many mutations. And because of that, most organisms have ways to kind of actually combat their activity that are sometimes similar to how to certain systems for combating viruses. However, there are definitely many instances where proteins that are beneficial or have been shown to evolve from transposons inserting themselves. So like I think it's kind of, they're a driver of mutation, and that when out of line can be negative, or it just can be negative in general, I guess, but also can be positive in the very rare circumstances that you get something beneficial from it.
Do you have an example of kind of a really bad trait that resulted from a bad mutation from transposons?
That's a really good question, and I know that they have happened, because I remember when I was researching them for like the first time, I came across several papers where people had found a disease in a particular person that was caused by a transposition event.
I was going to ask if you knew how that would be related to CRISPR gene editing technology. Does CRISPR use retrotransposons?
No, it doesn't. It's not actually that related. So
Mhm.
CRISPR-Cas9 is basically, it's a bacterial immune system that we took and we're like, we can use this in cells that aren't bacteria. So how that it works is pretty much when a bacteria infects a cell, when a virus infects a bacterial cell, one of the mechanisms that a bacteria has to fight it is to have all these little tiny DNA sequences that match with viruses that have infected, I think it's ancestors. Yeah, it's ancestors, and so if like a little bit of that sequence matches, it will bind to it, and then a protein called a kinase, it's basically like molecular scissors that come in and just chop up the DNA, will find that little tiny bit of bacterial DNA that matched with the viral DNA and just cut it to bits. And so what that we did is we're like, well, if we have this kinase that finds that can find this little any kind of DNA strand that has this little marker on it, and just target that one place and create a cut in it, then we can use that and give it, little chunks of DNA that are in areas of like other organisms that we want to have cut out. And so then we can tell that kinase to go in. So we'll send in the little DNA first. It will bind to where it needs to go. And then we'll send the kinase in and we'll snip right there. And then we can cut out that chunk.
Yeah, so it corrects genetic defects and all kinds of things, right?
Yeah, so we've kind of co-opted it to use it for that. And it's not really, I think it's just also used where if there's like, you're like, I want to know what this gene is, then you can use CRISPR and just cut it out and then compare it to the function of a normal cell, that hasn't had that part cut out. And you're like, what's different? And then it can kind of help tell you some information about what's going on,
Yeah.
I think the thing about CRISPR that's cool is, in my understanding, in terms of genetic modification techniques, it's pretty easy. I also know very little about it. My description was really bad, and people out there who are interested should read about it, because it's like both terrifying and like kind of ingenious.
I mean, essentially, it sounds like it's a like DNA scissors.
Yeah, DNA scissors that are powerful because you can choose where you want them to cut. So it's like, I think the thing about it in terms of what's scary when people think about like genetic modification is just the biology is so complex that sometimes you don't know everything that's going to happen, from removing one single thing. Like if you've ever seen a map of protein interactions, you can have one protein and it's just circled by 1,000 other proteins with little lines going to it. And then each of those is like circled by like just so many other proteins that it kind of creates this almost, this web that's really hard to pick apart and understand. And so, you can definitely mess with it and see what happens.
Wonderful. Well, thank you so much to Xander and Payce for joining us, myself and Bernie, on Scientifically Speaking here on WMPG. Stay tuned for songs from Power Out with DJ Cole. And from your favorite nerds, we wish you safe viewing and clear skies.
Will I die? Burn it. The moon shines pretty bright. Burn it. But I'll always remember you're my guy. Burn it. Tell me what's going on. Burn it. We just want to know where the asteroids are. The asteroids are flying around. You need to go and shoot them down. Barney, galaxy defender of all
humanity.
Bernie.
Ryan.
What is the title of the song?
What's the what?
What's the title of your song?
Oh.
We just want to know where the asteroids are.
Xander just said Bernie Licious.
Asteroids are flying around. Need to go and shoot them down.
Oh yeah,
Barney,
there's a part where he shoots asteroids out of the sky.
Wait, is this a song about Bernie?
Yes, yeah. I don't know if you heard the lyrics. They're kind of hard to
I couldn't hear them that clearly.
Okay.
Yeah, I didn't hear that clearly either.
Yeah, it's about Bernie shooting asteroids out of the sky.
Oh, okay.
Yeah, this is the third song we've written for Bernie. The
Okay.
last
Aww.
the last two we performed live in front of a lot of people.
Yeah, yeah, that was fun.
You know,
In front of the class?
Yeah,
well,
at the school. We used to do a thing at the school, the new school.
Yeah.
Oh.
everyone in the whole.
There was a line in it which was, Bernie could lift the sun, provided he had a very long lever.
Yeah.
Do
Okay.
you have the lyrics written down for this one?
We don't know.
No, it was literally, I was banging on the fridge and Payce started singing about Bernie. So, it was
It's just-
Do you expect that they will eventually be, like, we will evolve to a point where we don't have any more?
That's a really good question. I think every organism has a certain amount of pressure to kind of fight them. And a lot of them do disappear just kind of through genetic drift. But like, as you can see by looking at the history of our own genome, where you can see lots of them that are no longer active. I think one of the things that's kind of interesting about horizontal gene transfer and retrotransposons is that it's kind of been argued as a strategy for the retrotransposon to avoid that almost, because sometimes ending up in a new organism can avoid some of it can be less regulated in like a new host. And I think it's kind of interesting even within our cells, where like what you see is the only times that they actually are able to functionally really be retrotranscribed is when that cells are really when the cells are replicating lots of DNA. So, and like doing it in a way that's really unregulated, which is basically like the early embryo in cancer. And so like cancer cells actually have really high rates of retrotransposon activity, and so do early embryo cells.
What would you recommend, something that we did not cover on here, what would you recommend to a listener to look into more?
Let's see. I don't know. What I'm currently nerding out about, or was like up until maybe I'm finding my newest weird interest, is a thing where some anaerobic microorganisms are able to grow inside of tumors, because tumors usually, with the way that they grow, end up with pockets that have very little blood flow and consequently very little oxygen. And so you end up with kind of colonies of bacteria in tumors that can't actually survive in your bloodstream, and often they're gut bacteria. And there's interesting theoretical treatments being designed that use some of these bacteria. That's a really interesting subject to me. But
I felt like that was like a cliffhanger though, because you know, it's like, here, there's this thing, but find out more on your own. No, I love it.
In terms of retrotransposons, if any of you want to find interesting, if anybody else there wants to find interesting events where people think that horizontal gene transfer has happened and then write them in, that would be awesome. I don't know if there'd ever anything be done with them, but I'd like to see what people find. Just before I was talking with you, I was reading a paper on horizontal gene transfer and retrotransposons in plants, and I don't know, it's pretty cool. So. I
That's
think
awesome.
This is how they work as I understand them. Now, so we start with the, what's it called? The genomes. And inside the genomes, we have, what are they called?
Gene?
The genes. Hold on, let me start again. I forgot the essential word. That was the complete end of this. DNA. Okay, let me start again.
DNA makes up the never mind.
Okay, so let's think about this one more time. I'm going to introduce this. Okay, so.
I don't know.
Cool.